Hello student, once again welcome to Anu's classes. Today I am going to discuss another previous year question paper, that is question paper of 2017, and first part it is the botany section. So first question is, who is regarded as the father of green revolution? The answer is M.S. Swaminathan. Second is, the pollination done by snail is called Malacophilus. What is GMO? Genetically modified organisms are those in whose DNA from the desired organism is inserted in the laboratory. Next question. Name the bacterium which was controlled with penicillin by Alexander Fleming. An example is Streptococci bacteria. What are the disadvantages of asexual reproduction? The asexual reproduction, when only single parent is involved, that is reproduction is called asexual reproduction, and these are the disadvantages of asexual reproduction: lack of diversity. And the second point is, since only one parent is involved, therefore diversity among the organism is limited and they are unable to adapt the changes in the environment since single parents is involved. And last point, a single chances of in the environment may eliminate the entire species. Next question, with this question another or is coming, that is define parthenogenesis. Give an example of natural parthenocarpic fruit. Answer is, parthenogenesis is a type of asexual reproduction involving the development of female gametes without any fertilization. Example is banana. What is biofortification? What are the objective of it? 1 plus 1. So biofortification is the process of improving the nutritional quantity of food. And what are its objective? Or if question come, what are its significance, then also you have to write the same answer. The significance or objective is to reduce the mortality and morbidity rates related to the micronutrient malnutrition, and to increase the food security, productivity, and the quality of the life for poor population in developing countries. Or another question is given also, that is, what is selection? Name to method of selection when individual plants or groups of plants having the desired characters are picked up from population, eliminating the undesirable one is called selection. The two methods of selection are mass selection and pure line selection. Next question, what is transgenic plant? Give two examples. Transgenic plants are those whose DNA is modified using the recombinant DNA technology techniques. Example is BT cotton. Next, or is, what is recombinant DNA? How does enzyme endonuclease help its formation? Recombinant DNA is the artificially created DNA which incorporates DNA segments from different sources into one. A restriction enzyme is a protein isolated from bacteria that clips means cut down the DNA sequence at sequence specific site, producing DNA fragments with a known sequence at each end. Next question, give the full form of ELISA. Name two diseases for the detection of which this technique is used. And full form is enzyme linked immuno assume. And HIV, who is cause AIDS and lymph disease and the rotavirus disease are detected by the help of this technique. Next question is, what is E. coli RI? Mention its function. It is a restriction enzyme or restriction endonuclease. It cuts the DNA double helix at specific site. This restriction enzyme was first isolated from E. coli. That's why this name is given. They clip the DNA at specific site, the site known as the restriction site. The restriction enzymes protect the life of the bacteria from the bacteriophages. They recognize the cliff at the restriction site of the bacteriophage and destroy its DNA. So if question carry 2 marks, so then no need to write so much. If sometimes this question carry 3 marks also in previous year question paper, when I have analyzed, I noticed. That's why I'm giving the answer in long way. It is answer for 3 marks. If 2 marks come for the second para, you just write up to here only. Next question, write short notes on any one. First one is biopiracy. Biopiracy is the use of wild plant by international companies to develop medicines without paying any money to the countries from which they are taken. It is being carried out by many organizations and multinational companies by posing that components of the resources are product of their research. Example, turmeric, neem, etc. Next, gene cloning. DNA cloning is the process of making multiple copies of particular segment of DNA. During this technique, the selected DNA fragments is inserted into a plasmid using enzyme. Restriction enzyme and DNA ligase are used in this process. The restriction enzyme are used to cut the DNA fragments as specific sequence, and DNA ligase enzymes are used to join the nicks. Next, how insulin is produced through genetic engineering. Genetic engineering, you know, the process through which the improved quality of animals or plants are formed. That is called genetic engineering. So, the steps for production of human insulin is first, human insulin is extracted from the pancreas of the cells. Then, in a plasmid DNA, which is extracted from bacterium, cut and restriction enzyme and forming the plasmid vector. Then, the inserted human insulin is producing gene into the bacterial plasmid vector to form the recombinant DNA of the human insulin producing gene. Next, introduce the recombinant DNA into another bacterial cell to form the recombinant bacterium. And the recombinant bacteria multiply and produce the human insulin, and later the human insulin is extracted, purified and bottled. It is then ready to be injected to the diabetic patient. Next question, explain gene therapy by citing the example of ADA deficiency. Gene therapy is the process of introducing DNA into an organism in order to treat a disease. It is used to replace missing gene product or to correct the mutant alleles. ADA is an autosomal recessive inheritance disorder that occurs due to the defective adenosine deaminase enzyme. So this is the answer. If 2 marks come, you just write up to here. If this question carry 3 marks, then you will write the later portion. Next, describe the process of development of male gametophyte in angiosperm with suitable diagram. So, microspore or the pollen grain represent the initial cell of the male gametophyte. So, here I am showing in explanation is given. A diagram is this. Diagram means this is the mature anther. From here, the male gametophyte are released. In this microspore, first was there was the outer layer is called exine, inner layer is called intine. In the center, nucleus is present, and this is the germ pore. Germ pore means the portion where the exine is absent. In the next stage, these are the vacuoles, and this is the nucleus. In the next stage, the nucleus is divided into two cells. One is called vegetative cell, another is called generative cell. Next step, the intine form a pollen tube through the germ pore, and the two both the vegetative and generative cell enter into it. Next, as the pollen tube increase, the generative cell undergo division and it divided into two male gametes. And finally, in the pollen grain, there are two male gametes and one vegetative cell or tube nucleus. So this is the development of a male gametophyte. What are the advantages of cross-pollination? So there are, you know, the two type of pollination, self-pollination and cross-pollination. So cross-pollination, these are the advantages. So these are the advantages of cross-pollination. Here I am giving you 4 advantages. Next, what do you mean by GEAC? GEAC stands for Genetic Engineering Approval Committee. And what are the objective? And these are the objective of GEAC. Means to test the GMO product before releasing the market, and how it control the genetic engineering research to control the use of GMO crops in the field without a harm to the environment. These are the objective, or how it control the genetic engineering research. Answer will be the same. Or what is the full form of PCR? How amplification of DNA is done by using PCR. PCR means polymerase chain reaction. This is a technique used by amplifying small segment of DNA. So, and in this process, there are steps. Are first step is denaturation. Second step is called annealing, and third step is called elongation. So in the first step, what happened? In the first step, when the reaction mixer is heated at 94 degrees centigrade for approximately 0.5 to 2 minutes. And as a result, the hydrogen bonds are break between which join the two strands of DNA. In next is the singles. Now after the hydrogen bond break, the single strand now act as a template for the production of new strand of DNA. In the next step, the temperature is lowered to 54 to 60 degrees centigrade, where in the first test it was up to 94 degrees centigrade, but in this step it is lowered to 54 to 60 degrees centigrade for around 20 to 40 seconds. And here the primers bind to their complementary sequence on the template DNA. The primers are single strand sequence of DNA or RNA around 20 to 30 base in length. In one mark question, it may also come what is the primer in in PCR technique. The primer which is used, it has how many number of bases, then you have to write 20 to 30 bases in length. The two separated strands run in the opposite direction, and consequently there are two primer, a forward primer and a reverser primer. Next stage is elongation. Here again the temperature is raised to 72 to 80 degrees centigrade, and as a result the bases are added to 3 days end of the primer by the Taq polymerase enzyme. So name of the enzyme is important. You have to remember the name of the enzyme. For one mark question also it is important that it is elongates the DNA into 5 days to 3 days direction, and then these three steps are repeated 20 to 40 times in order to obtain a number of sequence of DNA of interest in a very short period. And this is the diagram. First step is denucleation, then is allelic, and last one is extension. So look first in the first stage, by using the high temperature it is break and singles that both the strands are separated. Next step, this is the primer is attached with the template DNA, and then that primer is attached with the template DNA in the 3 days direction, and then it grow, and this growth is from 5 days and to 3 days end. Describe the application of microbes in sewage treatment. So these are the application of microbes. So nothing to explain here. Or next is state the utility of the following plants, mention their scientific name. So 3 names is given. First one is Ashwagandha. So here the scientific name is given. That is, next is this is the medicinal use or economical use of the Ashwagandha. Second one is neem. This is their scientific name, and these are the economical use. Next, third one is Gomari. In common English language, common English name is Hiltic, and the scientific name is this one, Gamelina arborea, and this is the economical use or utility. Describe the fact in which Darwinism theory is based upon. So this question is carrying on 5 marks from evolution chapter, and this is Darwinism means the theory of evolution by natural selection as formulated by Charles Darwin book on the origin of species, which was published in 1859. So this year is also important for 1 mark question, and 4 key points of Darwin theory of evolution are what? These are the key points. So these are the key points. Individuals of a specific are not identical trait are passed from generation to generation. More offsprings are born than they can survive, and only the survivor of the competition for resource will reproduce. Or describe the factor involved in the process of organic evolution. So these are the factors which are involved in organic evolution: heritage, mutation, selection. These are the factors. Drift. These 4 are the main factors. Write a note on the abiotic factor influencing the growth and development of plant. So abiotic factor means non-living factor, which are temperature, air, water, soil, etc. So these are the effect of abiotic factor. First one is temperature, second one is water, third one is wind, fourth one is light. Or what is tissue culture? How does it help in crop improvement? Tissue culture is a method of biological research in which fragments of tissue from an animal or plant are transferred to an artificial environment in which they are continue to survive and function. And this is the rule of tissue culture in crop improvement. Excuse me. So this is the botany part of the 2017 question paper. Now come to the zoology section. Zoology section, so first fill in the blanks will come. So this 4 are the fill in the, 4 is the fill in the blanks, out of which you have to, in exam, student have to write 2 answers. First one is primary food plant of silkworm, answer is mulberry. Next one is animals that have had their DNA manipulated to express an extra gene are known as transgenic element. The process of release of ovum from the mature follicle is called ovulation. and group of individuals in a well-defined geographical area share or compete for similar resources potentially interbreed constitute the population. Next, one more question. Any two you have to write in exam. So what substances are present in birth control pill? You have to remember human-made estrogen and progesterone hormones are present. Next, which sex organ is present only in male? Answer is testis. What is the equation of versus per logistic growth population? So this is the equation, this growth, but which maximum value of the population. Which genetic code is said to be degenerate? So this is the answer for this. Because a single amino acid may be coded for more than one, that is the main reason of the answer. Answer any for the following, each is carrying 2 marks. So for these questions, I am giving you the answers for 2 marks. Write the location of the UTR in the mRNA and state its function. So this is the location of the UTR. First you have to write, then you have to write its function. So this is the answer. How the seminal plasma is formed? Write one function. So seminal plasma means a secretion of the seminal vesicle, prostate and bulbouterine gland, means all the 3 glands secretion when combined it from the seminal plasma. And what is its function? This is its function. Next, write how the sex is determined in man. You know man has the X and Y chromosome. So this is the process of sex determination mechanism in man. Next, describe briefly the structure of nucleosome. A nucleosome is the structural unit of DNA packaging in eukaryote. And this how sex is determined in man. If this question carry only 2 marks, then you have to no need to only you just write the notes. And if this question carry 3 marks, then with this note you just draw the diagram also. Next, what is the autoimmunity disease? Give an example. It is a disease which body's natural defense system cannot tell the difference between the body's own cell and a foreign cell, cutting the causing the body to mistakenly attack the normal cells. Means body's immunity system cannot determine which is the actual which is the body cell and which is the foreign particle. Next is name the disease of silkworm caused by protozoan parasite. Mention the prevention measure. So paper disease is of silkworm, and this is the preventive measure. State the difference between any two DNA, RNA. So this is the difference between DNA and RNA. This answer you get in class 11 also. Next, difference between acquired and innate immunity. So this is the acquired immunity and innate immunity. Difference. Next differentiation is production and decomposition. So this is the difference of production and decomposition. Next, this is the difference between two genetical disease. So these are the differences. Next question is coming. Draw the level diagram of a molecular structure of human fetus in uterus. So structure of human fetus within the uterus. So you have to label here uterus wall mainly and the fetus. Draw the level diagram of the molecular structure of antibody. So this is the structure of antibody. This is the antigen binding site and this is the antigen binding site. This is the variable constant and this is the light chain and this is the heavy chain. Write briefly the importance of beekeeping. So this is the answer, means advantage of beekeeping or the importance of beekeeping. Next, why should conserve biodiversity? So this is biodiversity conservation is why important. That's the answer is given here. A child has blood group O and if the father has A and mother has B, what will be the genotype of parents and the possible genotype of other offspring? So a child has O blood group, both parent has, means at least one copy of I allele, because when both the allele is I, then only the blood group will be O. So this will be the cross. Suppose father is I and mother is, father is A, mother is B, then father should have one A allele and one I allele. Mother should also have one B allele, one I allele. Then the next offspring, one will get IA and IB, and his or her blood group will be AB. Next one has blood group A, next one has blood group B, and last one has both the I allele. That's why this child will be have means O blood group. Explain codominance with example. Codominance means the phenomena of in which two alleles are expressed to an equal degree within the organism. So in offspring, both the alleles can express itself equally. And best example of codominance is ABO blood group. What is the infective stage of malaria parasite? Describe the life cycle of malaria parasite in brief. So the stage of infective for human is uninucleated lancet-shaped sporozoid. Sporozoids, and this is the life cycle of malaria. So first, what happened? First, look, formation of male and female gamete within the red blood cell. You start from here. So suppose this is the malaria. When the means mosquito bite the human, then the sporozoids are injected to the human blood with bite of female and Anopheles mosquito. Next is the sporozoids reach to the liver through the blood and reproduces asexually. In next step, bursting the liver cells to release the parasite into the blood. Next, the parasite reproduces asexually in RBC and causing the cycle of fever. In next stage, formation of male and female gametes in the RBC. Means within the RBC, some means sporozoids divided two gametes. One is male gamete, one is female gamete. And then if that patient is bite by any mosquito, then the female mosquito take up the genotype with the blood meal. Means when it means take the blood as meal, the male and female gametes of the disease also enter into the mosquito's body. Then fertilization and development takes place in the intestine of the mosquito. Means sexual reproduction takes place within the body of the mosquito, whereas asexual reproduction takes place within the body of the human. Next, mature sporozoids migrate into the migrates from the intestine to the salivary gland of the mosquito and store there. And when next it bites human beings, then it will go for the human body and the cycle will be repeated. What are the possible causes of cancer, and write briefly on its detection and diagnosis. There is no single cause for the cancer because scientists believe that it is the interaction of many factors together that produce cancer. The factor involved may be genetic, environmental, or constitutional characteristic of the individuals. And there is no single test that can accurately diagnose cancer. The complete evaluation of a patient usually requires a throughout history and physical examination along with the diagnosis testings. Many tests are needed to determine whether the person has a cancer or if other conditions such as an infection is mimicking the symptoms of cancer. Imaging tests used in diagnosis cancer may include the computerized means CT scan and bone scan, magnetic resonance imaging means MRI, then PET test, ultrasound, X-ray, among others. Biopsy. During a biopsy, doctor collect the sample of the cells from the testing in the laboratory. Next, what are the different stages of gene regulation? Explain the lac operant model of transcriptional regulation. So in eukaryotes, gene regulation occurs at any of the following step: transcriptional level, that is during the formation of primary transcript, processing level, that is at the stage of splicing, during transport of mRNA from the nucleus to the cytoplasm, translation level. In the lac operant, lactose acts as an inducer, and if the lactose is provided in the medium for the bacteria, the regulatory genes are activated and the inducer will bind to the receptor protein and render inactive, which allow the transcription of the operand. Thus, the lac operant is negative regulator in this case. And this is the diagram. Suppose lac operant model. First, look, lactose is absent. Then what happened? This is the regulatory gene. Because the lac operant has promoter portion, then structural gene portion. Then this is the regulatory gene, and this regulatory gene transcription, by the process of transcription, it from the repressor mRNA. And this is a repressor. Then this repressor, if the lactose is absent, then what happened? It will bind this portion, and as a result, the no transcription process can take place here. But if the lactose is present as inducer, then what happened? When the repressor is produced, the repressor is become inactive by the use of this inducer, means lactose. And as a result, what happens? It cannot bind at this site, means an operand, it cannot block the operand, and the transcription process will be continued. So the LAC operand work as a switch on and switch off for the transcription process. Next, explain the Southern blot hybridization tactic for DNA fingerprinting. So this is one of the important questions. The Southern blot is a method used for molecular biology for detection of a specific DNA sequence in a DNA sample. Southern blotting combines the transfer of electrophoresis separate DNA fragment to a filter membrane sequence and fragment detection by the probe hybridization. So this is the flow chart of the technique. So in exam, you just write the flow chart, then also you will get the full marks. No need to write in long parawise. So first isolation of DNA takes place, then diagnose the DNA by this enzyme. Next, separation of DNA fragment by electrophoresis, then transferring of the separate DNA fragment into the synthetic membrane such as nitrocellulose or nylon. Next, hybridization of the using level VNTR probe and detection of the hybridization DNA fragment by autoradiography. And this is the diagram of the process. First, this is the DNA. After restriction enzyme, by the help of restriction enzyme or molecular size, it is cut into pieces. Then they are taken the gel, and after that it transferred to the means paper towel. It is transferred into the, with the help of paper towel, it is transferred into the membrane gel, and then by the process of probe DNA, they are diagnosis and detect the required DNA probe. So this is the process of excuse me, yeah, process of Southern blot hybridization.